This
Good morning,
conference
ma'am.
will now be recorded. This conference will now be recorded.
Good morning.
So yesterday we had finished with transcription in the prokaryotes, and we have seen that for the transcription to occur in the prokaryotes, what we require was a coding stand, a template stand, and the RNA polymerase enzyme, which consists of a core enzyme and a sigma factor. That is very much essential to recognize the promoter sequences. So this promoter sequences were, or for initiation of the transcription, were the data sequence that was 10 base upstream of the transcription site, and a 35 base sequence that was 35 base upstream of the transcription site. And it had transcription involved initiation, elongation, and termination. So the pre-initiation complex was formed by the sigma recognizing the promoter sequence, and in turn again attaching the RNA polymerase to the transcriptions or to the initiating size, and then there was a formation of a pre-initiation complex. So in the initiation complex, there was attachment of the template, and then RNA polymerase was attached to it after the sigma factor recognized the bubbles, and then formation of the pre-initiation complex. With later on, there was a release of the promoter clearance with the release of the sigma factor, and the RNA polymerase moved to, after synthesizing 15 to 20 nucleotides, and moved on to the next phase of next part of the DNA to synthesize the RNA strand again. So we will now see what is the difference compared to prokaryote. What is the differences here in the eukaryotes? So the eukaryotic transcription, it is more complex, slower, and more flexible than in prokaryotes. And this comes at the cost of greater complexity and the time that is needed to make a protein. So here you can see this is the prokaryotic, where from the DNA synthesis of RNA and mRNA and ribosomes, that is, this is the cytoplasm, while here the processing is more in case of the eukaryotes. So what are the differences in the prokaryotic and eukaryotic transmission is that there are 5 eukaryotic RNA polymerase are there, and they are core promoters on the eukaryotic DNA. Other than the promoters, there are also transcription factors. They are required for the RNA polymerase. And RNA polymerase, DNA binding, or you can say for transcription. Then other than this, there are also cis elements, enhancers or repressors. And then termination of RNA polymerase or transcription will take place. So what are the more additional things we have got over here? Other than the promoters, we have the transcription factors, we have the cis elements, we have the enhancers or the repressors. And termination again here in the eukaryotes will be different from that of the prokaryotes. Now we said here there are 5 types of RNA polymerases here. So the first 3 RNA polymerase 1, 2, and 3, they are nuclear RNA polymerases, while the 4th one is a mitochondrial, and the 5th one is a chloroplast RNA polymerase enzyme. Now each of these DNA dependent RNA polymerase, they are responsible for the transcription of different sets of genes. Now this nuclear dependent RNA polymerase, they exhibit more complex subunit profiles than the prokaryotic RNA polymerases. And they have 2 large subunits and a number of smaller subunits. The functions of each of these subunits is not yet fully understood. So we are dealing now mostly with this 3 polymerases, 1, 2, and 3. You can see that RNA polymerase 1 is responsible for the production of ribosomal RNA, while 2 is responsible for the production of messenger RNA, mRNA, and microRNA, and small nuclear RNA. While the third one, RNA polymerase 3, is responsible for transfer RNA, and for 5 is ribosomal RNA. Now all these 3 forms of RNA polymerases, Here, the sensitivity had been shown to amanitin. This amanitin is a peptide toxin that is produced from the mushroom. So mushroom, the scientific name for the mushroom is the Amanita phyloides, and it produces this peptide toxin. So this we have seen the sensitivity of this RNA polymerase to this peptide. So you can see the type 1, it is insensitive, while the type 2, which synthesizes the mRNA, it is very highly sensitive to this toxin, while the type 3 shows intermediate sensitivity. Now, for transcription to occur in the eukaryotes, a core promoter must be present near where the RNA transcription is to begin. And there are many types of promoters will be seen in the eukaryotes. And each type of RNA polymerase, either 1, 2, and 3, they will look for its own unique promoter. And they will synthesize its unique RNA product type. So, RNA polymerase 1, we said, is synthesizes what? Ribosomal RNA, 2 mainly messenger RNA, and than that microRNA and small nuclear, messenger RNA and micro and small nuclear RNA, and 3 will synthesize the transfer RNA. Now, what are these eukaryotic promoters? So you can see here, this if this is the transcription site over here, this what we say is are the upstream elements. So this is upstream to that of the transcription site. This site will be the downstream to that of the transcription site. So here the main eukaryotic promoters, they are in the Tata box like you have seen in the prokaryotes. But here it is located only 25 to 30 base upstream from the transcription start site in the mammalian genes that contain it. So that was 10 base upstream. This is 25 bases upstream to that of the transcription site. Then this starter box also here is known as the Hogness box, and the sequence is TATA. Then 2 other types of sequence elements will be seen. They are known as promoter proximal upstream elements. So they are proximal to that of the promoter elements. That's why they are known as promoter proximal upstream elements. So what are these 2 types of sequences that you see? This is GC and CAT boxes. So you can see GC and CAT boxes will be these other promoter proximal upstream elements located 70 to 80 nucleotides upstream from the start of transcription. Then other than this, also sometimes what happens, some number of the mRNA encoding genes, they may lack a consensus starter box. So in this case, when mRNA lacks this starter box, so you need something for the as a promoter. So in such cases, additional cis elements and initiator sequence, this is the INR here, INR, or a downstream promoter element DPE, they will direct the RNA polymerase transcription machinery to the promoter, and so they will be providing basal transcription starting from the correct site. So this elements will help whenever the mRNA coding genes they lack the data sequence. Then together the promoter and the proximal cis active upstream elements, they will confer fidelity and frequency of initiation upon a gene. So here the enzyme itself is not concerned with the fidelity. Here it is, these are the elements, promoter proximal elements that will confer fidelity and frequency of initiation upon a gene. Then a third class of sequence elements that can either increase or decrease the rate of transcription initiation of the eukaryotic genes, they are called either the enhancer or repressors, or otherwise they are silencers depending on how they affect the RNA synthesis. That means the enhancers will increase the rate of transcription initiation, and repressors will decrease the rate of transcription initiation. So now what are the types of promoters we have seen? We have seen the Tata of the Hogness box. We have the promoter proximal elements GC or CAT sequences with a 75 to 80 base upstream. And then we have the enhancer and the repressor elements. Other than that, there are also other regulatory elements, but we won't go through that now. So all these are called as distal regulatory elements. So we have promoter elements, promoter proximal elements, and distal regulatory elements. And this INR and DPE and they are cis elements, they will come into a picture whenever the Tata box is not there in the mRNA coding genes. So the RNA polymerase enzyme, that is either 1, 2 or 3, they will transcribe the information that is contained in the template strand of the DNA into the particular type of RNA that they are going to produce. Now these polymerase now, in order to begin the transcription, they must recognize a specific site in the promoter in order to initiate the transcription at the proper nucleotide. Now, in contrast to the situation in prokaryotes, the eukaryotic RNA polymerase, here they are not alone, they are not capable of discriminating between the promoter sequence and other regions of the DNA. There in prokaryotes, what it was the sigma factor that recognizes the data sequence. But here the RNA polymerase by themselves, they were not able to discriminate between the promoter sequence or the other regions of the DNA, and they require help. So other proteins that are known as the general transcription factors or the GTFs, if you want to say, they facilitate the promoter specific binding of these enzymes. So these transcription factors will help in for the RNA polymerase to recognize this promoter sites and formation of the pre-initiation complex. Now they are these transcription factors, they are 6 in number. So you have transcription factor 2A, 2B, D, E, F and H. So there are 6 transcription factors. Now this out of this transcription factor 2D plays a very important role here. It consists of a 34 kilodalton Tata binding protein TBP and 14 non-TBP protein subunits that are known as TBP associated factors. So they are 14 in number. So this complex of TBP and TAFs together, they form the transcription factor 2D. Now binding of this transcription factor 2D complex to the Tata box sequence through this mainly the Tata binding protein is thought to represent a first step in the formation of the transcription complex on the promoter. Now it has been postulated that these transcription factors will bind stepwise, or first they may bind to each other to form a complex and in turn to the enzyme RNA polymerase. So 2 models have been used to explain the assembly of the pre-initiation complex. Here you can see this is one model and this is another model. So this is a stepwise model. This is by forming a whole enzyme. Here you can see all the transcription factors as bound together to the RNA polymerase, and then they are forming the pre-initiation complex. They will bind to the promoter sequence and form the pre-initiation complex. While here, if you see in the stepwise manner, what is happening is that here the sequential binding of the transcription factors is taking place. So here, first the transcription factor 2D will bind to the Tata sequence over here, and then the DNA binding transactivator proteins specifically bind enhancers and in part facilitate the pre-initiation complex formation. So these enhancers are also going to help in the formation of this pre-initiation complex by binding directly to the transcription factor 2D and transcription factor TBP association factor subunits. So here, so first is the binding of the transcription factor 2D to Tata. Once this binding this transcription factor 2D recognizes and binds to the Tata sequence, this is followed by addition of transcription factor A and transcription factor B. Now, once this complex has formed from transcription factor 2D, A and B, this in turn, they will bind to RNA polymerase. they will attract the RNA polymerase and transcription factor F and bind to them or tether to them bind to them these two polymerase and two transcription factor F. Later on, this transcription factor H and E will join in the complex. So all this together, they form the pre-initiation complex. So we'll just see what we have just seen in the figure. The binding of transcription factor 2D, it marks a specific promoter for the transcription. Of the several subsequent in vitro steps, the first is the binding of transcription factor A, then B to the transcription factor 2D promoter complex. So this will result in a stable ternary complex, which is more precisely located and more tightly bound at the transcription initiation site. Now this complex that is composed of D, 2AB and the promoter sequence, they will attract and tether the polymerase 2 enzyme and the transcription factor 2F complex to the promoter. When I'm talking about polymerase 2 enzyme, it means we are talking about the synthesis of the messenger mRNA. So from the DNA, messenger RNA is being transcribed by polymerase 2 RNA polymerase enzyme. So this attracts and tethers this polymerase 2 and F complex to the promoter. So addition of then transcription factor 2H and E are the final steps in the assembly of the pre-initiation complex. So once all these come together, the pre-initiation complex is now complete and capable of basal transcription initiated from the correct nucleotide. So here you can see this is the Tata consensus sequence to which the transcription factor 2D is binding, followed by binding of A and B. This ternary complex in turn leads to binding of polymerase, attract the polymerase 2 and F, followed by H and E. So all these together are forming the pre-initiation complex. Now this entire complex spans the DNA from position -30 to +30, meaning this is upstream and downstream of the initiation site relative to the initiation site +1. That is this. So this is the initiation site. So this is the spanning from -30 to +30. Now this Tata binding protein, we said, which is a component of the transcription factor 2D, it binds to the Tata box in the minor groove of the DNA. While most of the transcription factors, they bind in the major groove, it's only the TBP will bind with in the minor groove. And this will cause an approximately 100-degree bend or kink in the DNA helix. So this you can see, this is the bend that is occurring in the DNA helix. This bending is thought to facilitate the interaction of the TBP associated factors with other components of the transcription initiation complex. The multi-component eukaryotic promoter and possibly with the factors bound to upstream elements. So this bending will help in more further interaction with all the other components of the transcription complex. So what are the function of this transcription factors? So we have already seen transcription factor 2 is the one that initiates and binds to the Tata DNA, and that is that is called as the TBP or transcription binding protein, and then it directs the assembly of several other components by protein DNA and protein-protein interaction, that is transcription factor A, B, E, F, and H, and polymerase 2. Then transcription factor A and B, they bind or modify the transcription 2D, while 2E binds and forms a pre-initiation complex. 2F will now bind at a Tata box near +1, that is the site of initiation. And while binding of H, transcription factor 2H permits DNA unwinding helicase and phosphorylation of the RNA polymerase 2. So phosphorylation will lead to activation of RNA polymerase 2. Now this eukaryotic polymerase 2 enzyme, it consists of 12 subunits. And there are two large subunits of 150 and 190 kilodalton, which are homologous to the bacterial subunits. And then in addition to the increased number of subunits, the eukaryotic polymerase 2 differs from the prokaryotic counterpart in that it has a series of heptad repeats with a sequence, consistent sequence of tyrosine, serine, proline, threonine, serine, proline, serine at the carboxy terminus of the largest polymerase 2 subunit. So this is called as the carboxy terminal repeat domain. So the importance of this domain is that the polymerase 2 is activated whenever when phosphorylated on the serine and threonine residues, and it displays reduced activity when the CDD is dephosphorylated. So this heptad subunits are present at the carboxy terminal repeat domain, and phosphorylation of the serine and tyrosine residues will lead to activation of this polymerase 2 enzyme. Dephosphorylase will inactivate the enzyme. So CTD dephosphorylation, they are critical for promoter clearance, for elongation and termination, and even for mRNA processing. So up till now, what we have seen what are the different types of promoter elements that we can see. Promoter, proximal promoter and distal promoter regulatory elements. We have seen formation of pre-initiation complex, and then we have seen how the polymerase enzyme, it may be activated or inactivated. Then they are and now once we have seen this formation of pre-initiation complex, then synthesis of the RNA will take place similar to that of what we have seen in the prokaryotes. So once this pre-initiation complex is formed, then first nucleotide, which is most probably a purine, will be added, and the RNA polymerase 2 will lead to the synthesis of sequential addition of the nucleotide triphosphate, leading to synthesis of the heteronuclear messenger RNA. Heteronuclear RNA. Then specific signals, they regulate the transcription termination. The signals for termination of the transcription by the eukaryotic RNA polymerase 2 are poorly understood, but the explanation here that is given is that after RNA polymerase 2 has traversed the region of the transcription unit, encoding the 3' end of the transcript. So once the RNA polymerase goes on forming the transcript, hRNA transcript, then what will happen? This RNA endonucleases, they will cleave the primary transcript at a position about 15 bases, 3' of the consensus sequence. This is the AAU and AAA sequence that serves in the eukaryotic transcripts as a cleavage and polyadenylation signal. So whenever this sequence is encountered, about 15 bases, they will cleave at a position 15 bases 3' of the sequence. And this cleavage is occurring through the enzyme RNA endonuclease. So finally, you have this newly formed 3' terminal is polyadenylated in the nucleoplasm. So this we will see this about polyadenylation, we will see in the post-transcriptional modification. So once the primary transcript, that is heteronuclear RNA, is formed, in order to become the mature mRNA, it has to undergo extensive post-transcriptional modification. So the RNA produced during transcription, they are called the primary transcripts, and they are inactive. Now a group of enzymes, namely the ribonucleases, they will be responsible for the processing of transfer RNA. So whenever the transfer RNA is synthesized by the respective polymerase enzymes or ribosomal RNAs are formed, they undergo processing by ribonucleases in both prokaryotes as well as in eukaryotes. Now the prokaryotic messenger RNA, that was synthesized during transcription, they are similar to the functional mRNA. So they don't require post-transcriptional processing. So only the heteronuclear RNA or the primary transcript, the eukaryotes, they require extensive post-transcriptional modifications before they become functionally active. So what are these post-transcription modifications? We will see now. So alteration is occurring at the ends of the messenger RNA, at both the ends, at the 5' end as well as at the 3' end. So what is the alteration that are taking place? There is the addition of a protective cap at one end and a tail of adenine nucleotides at the other end. So what will happen if these changes take place? These both changes will have, that is addition of a cap or polyadenylate tail, will help to protect the RNA from enzymes that will cause its degradation. So what is this cap we are seeing is, it is a modified, in this, you can see in the figure here, it is this capping that is occurring at the 5' end. It is a modified guanine nucleotide that is added at the 5' end. And what is this modified guanine nucleotide? You will see in the next figure. And while at the 3' end, you have a poly A tail. Poly means there are so many adenine nucleotides. About 50 to 250 adenine nucleotides will be added at the 3' end. So addition of this 5' cap and poly A tail will prevent the mRNA or primary transcript from degradation by the enzymes. So the capping is what we are talking about is the 5' end of the mRNA is capped by 7-methyl guanosine. So it's a 7-methyl guanosine and by an unusual 5' or 5' triphosphate linkage. So here you can see this is a unusual 5' or 5' triphosphate linkage, that is triphosphate linkage that is seen, and this methyl group donor is through SAM. S-adenosine methionine is the donor of the methyl group over here. Then the poly A tail, we said they are around 52 to 15 adenine nucleotide that were added. This addition of this nucleotide is by the enzyme poly A polymerase enzyme. This poly A polymerase enzyme by hydrolysis of the molecule of ATP will add the tails at the 3' end. But this poly A tail will get reduced when the mRNA enters into the cytosol. So we have seen what happens at the end of mRNA. Then next is the removal of the intron sequences. I have already told you what are the exons and the introns. In the initial mRNA transcript, what you have the exons that are coding sequence. This is coding coding for genes that are intervened by non-coding sequences that are the introns. Now, in order to make it active, or mature, the introns have to be excised. So excision of these introns, how they are occurring is by these introns will be removed by a process that is called as splicing by small nuclear ribonuclear protein particles. That's also known as SNRPs, to produce the messenger RNA. Now, this what you mean by snRNPs are the small nuclear ribonuclear proteins. They are formed when the small nuclear RNA they associate with proteins. So when they associate with protein, they form small nuclear ribonuclear particles. These in turn will cause the splicing of the introns. So here you can see capping is taking place, removal of the introns occurring, and in the end result is the formation of a mature messenger RNA. Now, alternate patterns of heteronuclear RNA splicing will result in different messenger RNA molecules which can produce different proteins. So, alternate splicing results in mRNA heterogeneity. So, different sort of proteins may be produced by different patterns of splicing. But faulty splicing can result in diseases. So, some mutations, either lengthening or shortening of a protein, lead to some conditions, that is beta thalassemia. Now, post-transcription modification of messenger RNA will take place in the nucleus. The mature RNA, in order to perform its function, then enters the cytosol to perform its function, that is translation. Now, how does this messenger RNA from the nucleus, it enters into the cytosol? So it is entering into the cytosol through the nuclear pores. Nuclear pores will be present here. Once it undergoes maturation, through the nuclear pores, the mature RNA is delivered to the nuclear pores, dotting the nuclear membrane, where upon transport through the pores, the messenger RNA can be engaged by the ribosomes for the translation to take place. Now, this messenger RNA is also said to undergo editing. So, 0.01% of the mRNA undergoes editing. A very good example we can see is that with the apoprotein B gene, where conversion of the CCA codon that is normally present in that gene in mRNA into UAA may occur by the enzyme cytidine deaminase. So what happens when this occurs? Because of this, even though this protein is originating from the same gene, the liver will synthesize a 100 kilodalton protein, which is ApoB100, while the intestinal cells will synthesize the protein as a 48 kD protein, that is ApoB48. So the gene is the same, but the proteins that you are getting in the end are two different proteins in two different organs. So in liver, you have a 100 kDa protein ApoB100, and in the intestinal cells, you have ApoB48. This is happening because the formation of the termination codon UA from CA will result in editing of the RNA. Now, transcription may be inhibited by certain factors. This year, we are talking about the drugs here or certain toxins. Actinomycin D was the first antibody that was used for the treatment of tumors, and they inhibit the transcription by blocking. They bind to the DNA template and block the movement of the RNA polymerase. Then rifampicin, which is used in the treatment of tuberculosis and leprosy. It binds with the beta subunit of the prokaryotic RNA polymerase and inhibits its activity. Then alpha-amanitin, I have already showed you this effect of this alpha-amanitin in association with the 3 RNA polymerases. So this toxin is produced by the mushroom Amanita phalloides. It is the taste is very good, it's delicious, but poisonous because of the toxin. And we said the sensitivity is this DNA polymerase 2 is very sensitive because this toxin will bind tightly with this enzyme and will inhibit the transcription. Then this entity that's known as reverse transcription. This reverse transcription we have seen when we are doing with cancer also, where the retroviruses, they produce RNA as the genetic material, and the complementary DNA is formed from this RNA with the help of the reverse transcriptase and insertion of this complementary DNA into the host DNA. Now, when we talk about the RNAs, we have seen now mainly in regard to the messenger RNA, how the transcription or the post-transcription modification are taking place for the messenger RNA. But you have post-transcription modification even for the transfer RNA and the ribosomal RNA. So before that, we'll just see what the total RNA comprises of. Out of the total RNA that is present in the cell, only 4% of the total RNA forms the coding RNA, that is mainly the messenger RNA, while 96% of the total consists of the non-coding RNA. So what are these non-coding RNA? These are the transfer RNA, ribosomal RNA, small nuclear RNA, and also the small cytoplasmic RNA. Now, as you have already read the transfer RNA, it consists of around 74 to 95 nucleotides. This is also generated by nuclear processing of a precursor molecule that we have also seen a pre-transfer RNA. And then what is the function of transfer RNA is that it translates the information of the sequence of nucleotide in the messenger RNA into specific amino acids. And there are at least 20 species of transfer RNA corresponding to 24 amino acids, corresponding to at least one or several, but corresponding to 20 amino acids required for protein synthesis. And the primary structure of this transfer RNA allows extensive folding and intrastrand complementarity to generate a secondary structure that appears like a clover leaf. Now, what are the post-transcription modification of the transfer RNA here? So, the enzyme that is responsible for synthesis of transfer RNA is RNA polymerase 3. So, they make the transfer RNA that carry the amino acid to the nascent protein chains that are produced from messenger RNA on a ribosome in the nucleus. So, they have clover leaf shape and hairpins made possible by the RNA bases, forming a DNA-like double strand. Now, the RNA that is formed from from transcription, this is what is there, and this will undergo post-transcription modification to form the mature RNA. So, what is happening here? The pre-transfer RNA nucleotides are removed, replaced, and modified. So, you can see what is being replaced here. Replacement of the nucleotides here, you can see it is UUA. Here, replacement of the nucleotide occurs with CCA. And then you have the removal of the leader sequence from this end. Then chemical modification of the bases are taking place here. And excision of the introns, the purple ones where the excision of the introns will take place in this loop. So this is the primary transcript of the transfer RNA, and this is the mature transfer RNA. So all the transfer RNA molecule ultimately consists of 4 arms. You have the acceptor arms with the CCA, and these 3 nucleotides that are added. We said here the 3 nucleotides CCA that are added. They are added post-transcriptionally by a specific nucleotide transferase enzyme. Then the transfer RNA appropriate amino acid is attached or charged onto the 3' hydroxyl group of the A moiety, adenine moiety of the acceptor arm. Then you have the D arm, the T arm, and the extra arms that help to define a specific transfer RNA. And this transfer RNA roughly comprise about 20% of the total cellular RNA. Then coming to the ribosomal RNA, this ribosomal RNA, they are present in the cytoplasm, and they are responsible for the synthesis of proteins from the messenger RNA. Now on the ribosomes, the messenger and the transfer RNA will interact for the synthesis of the proteins. And during the periods of active protein synthesis, many ribosomes may be associated with any particular mRNA molecule to form an assembly that is known as a polysome. Now, these mammalian ribosomes, they consist of 2 major subunits, a 60S subunit and a 40S subunit. Then the 60S subunit again consists of a 5S, a 5.8S, and a 20S ribosomal RNAs, while the 40S subunit will contain only a single 18S RNA. Now, all this ribosomal RNA molecule, except the 5S RNA, which is independently transcribed, they will be processed from a single 45S precursor RNA molecule in the nucleolus. So we will see here what is happening for the post-transcriptional processing of the ribosomal RNA. So this pre-ribosomal RNA, the primary transcript is formed by RNA polymerase 1. So the 18S, 5.8S, and 28S RNA rRNAs, they are cut from a single larger pre-ribosomal RNA, and methylation will help to stabilize the structure. So you can see this is the pre-RNA 45S, and you can say these are the different ribosomal rRNAs over here. They will undergo cleavage to form the mature ribosomal RNAs. So you have the 18S, 5.8S, and 28S. Then next we have the small RNAs. Now most of the molecules are complex with proteins to form ribonucleoproteins, as we seen in the case of lysosome, and are distributed in the nucleus, the cytoplasm, of both. Now this small RNA vary in size from 20 to 300 nucleotides, and collectively they form roughly about 5% of the cellular RNA. Now this, what is the function of this small RNAs? They are involved mainly in the ribosomal RNA, mRNA processing, and as well as in gene regulation. Of the several small nuclear RNAs, some of them are important. So like U1, 2, 4, 5, and 6, they are involved in the removal of the introns and the processing of the messenger RNA precursors into the mRNA. While the U7 small nuclear RNA is involved in the production of 3' ends of the histone messenger RNA, whenever the messenger RNA will act a poly A tail, then it will add the histone to the messenger RNA. Then we have the microRNAs and small interfering RNAs. These two types of RNAs, they will cause inhibition of the gene expression by decreasing specific protein production, and it may be by different mechanisms. Now both these types of RNA, they are around 21 to 25 nucleotides in length, and both of them are generated by nucleolytic processing. But while microRNAs, they are the products of distinct genes or transcription units, while the small interfering RNAs, they are formed as a result of nucleolytic cleavage of larger double-stranded RNAs. So we have seen how the, if you want, we are going to summarize this, we are going to see that we have seen how the transcription of the eukaryote is taking place. In the transcription, when it differs from the prokaryotes, we have, beside the data of the Hogness box, we have other promoter elements, and the proximal as well as distal, that may be cis elements, they may enhancer elements are seen, and we have, in order to bind to the promoter site, it requires the help of the transcription factors. And there are six transcription factors. All these transcription factors as well as elements together, they form the pre-initiation complex. And later on, the elongation, synthesis of the RNA transcript and elongation will occur in a similar manner to that of the prokaryotes. Here, termination was with the help of an endonuclease, RNA endonuclease enzyme, by recognition of a particular sequence that is seen. And then later on, post-transcriptional, so the primary transcripts undergoes extensive post-transcription modification in the eukaryotes, and we have seen that there is a 5' capping and polyadenylate tail at the 3' end, then removal of the introns by splicing and formation of the mature messenger RNA. Then similarly, we have seen that post-translation modification also occurs in the transfer RNA, and we have seen that different removal of the leader sequence, changing in the bases in the loop, and changing of the sequence at the acceptor arm to CCA by the enzyme, that is the respective enzyme. And then in the ribosomal RNA also, we have seen how post-transcriptional modification occurs by splicing or cleavage of the particular RNAs. Then we have seen what are the function of the small nuclear RNAs that helps in the intron removal, and wherever the poly A tail is lacking in the messenger RNA. And then we have seen microRNA, small interfering RNA, and they help mainly in inhibition of the gene regulation.
Transcription is the process of making RNA from a DNA template. Several key factors are involved in this process, including DNA, transcription factors, RNA polymerase, and ATP. Transcription begins with a strand of DNA. It is divided into several important regions. The largest of these is the transcription. This portion of the DNA will be used to produce RNA. Upstream of the transcription is the Tata box. An enhancer region may also be involved. Several complexes, known as transcription factors, are required for successful transcription. The first is TF2D, the largest of the general factors. A component of this factor, TVP, binds to the DNA using the Tata box to position TF2D near the transcription initiation site. Other transcription factors, including TF2A and TF2B, then attach. These complexes prepare the DNA for the successful binding of RNA polymerase. Once RNA polymerase is bound, other transcription factors complete the mature transcription complex. Now, energy must be added to the system for transcription to begin. This energy is provided by the reduction of ATP to ADP and PI. RNA polymerase then synthesizes an RNA template stranded. Most factors are released after transcription begins. When the end of the transcription unit is reached, RNA polymerase dissociates, and the newly formed strand, RNA, is removed.